Hello students, welcome to Trunkie Academy. We are studying about the excretory system. Okay, in the last class yesterday I discussed about some basic concept and some basic topic that was about the uh, what is the role of kidney, how this uh, kidney plays an important role in the formation of urine. But the actual, the functional and structural unit of this kidney or the excretory system is the nephron. So that we will be studying in detail today. As I told you in the last class yesterday that there are different modes of excretion: ammonotelism, ureotelism, and uricotelism. Right? When this nitrogenous metabolic wastes are produced, okay, they are very toxic, very harmful, and must be removed and eliminated from the body as soon as possible. Ammonia is very dangerous and toxic, so that ammonia can be converted into some less harmful substances, less toxic substances called urea or uric acid. So that we find in case of some animals, like including human beings. Now we'll study about the structure of nephron, then how it looks like, what are the different parts of the nephron, and how it works in the formation of urine, okay, and what is the role in its role in osmoregulation. Okay, so it is the structural and the functional unit of kidney. What is the structure of nephron? It is looks like this. If it is a diagrammatic picture, okay, there are blood vessels, arteriole, thin-walled, coiled. The wall is very thin, it's coiled, and you can say some tubular structure, right? Lined by single layer of epithelial tissue. So this facilitates the movement of molecules. There are around 1 to 1.2 million nephrons in each kidney. Okay, so there are a pair of kidney on either side of the vertebral column at the lower abdomen. So here you can see that there are about 1 to 1.2 million nephrons in each kidney. Okay, that is the proximal end, and which is blind. Okay, proximal blind end, where this receives the blood vessels that brings the nitrogenous waste product along with the blood, and which is filtered there, okay, for the formation of urine. So the structure and functional unit of kidney is a nephron. I told you, nephron being the important part, or you can say can be grouped or divided into two parts: the Malpighian corpuscle and renal tubule. This Malpighian corpuscle again has Bowman's capsule and glomerulus, where the renal tubule has distal convoluted tubule, proximal convoluted tubule, loop of Henle, and the collecting duct. Okay, so you can see the nephron, this basic structural and the functional unit of this excretory system, or you can say nephron or kidney, can be divided into two main parts, that is the Malpighian corpuscle and the renal tubule. So if you observe the Malpighian, this Bowman's capsule and the glomerulus, they are together called Malpighian corpuscle. Okay, so in case of renal tubule, tubular structures are there apart from this Malpighian corpuscle, and that can be divided into proximal convoluted tubule, loop of Henle, distal convoluted tubule, and collecting duct. Right. So you can see the nephron. This is a longitudinal section. A proximal convoluted tubule starts from there. Okay, the capsule, capsular space is there, and then you can see this pedicel, the podocyte, the parietal layer, glomerular. Okay, glomerular Bowman's capsule, parietal layer of glomerular Bowman's capsule. Okay, that is the layer of the parietal layer. Okay, you can see the blood vessel, afferent arteriole, that is the blood vessel, and there is a juxtaglomerular cells, that are adjacent cell, that also plays a very important role in osmoregulation. Okay, so that is this is the one part. You can see the endothelium of the glomerulus. Okay, so Bowman capsule and glomerulus. Okay, so this is the Malpighian corpuscle. Okay, so this is the Malpighian corpuscle. As I told you earlier, both glomerulus and the Bowman's capsule, they together form the Malpighian corpuscle. And as you can see, there is a parietal layer of the Bowman's capsule. Okay, then the glomerulus, this part which has got the podocyte and the pedicel. Okay, these are the glomerulus, and the efferent arteriole and the efferent arteriole, bringing the blood and going, then taking out the blood back to the body. Okay, so that is the afferent and efferent arteriole. Afferent arteriole will bring the blood with nitrogenous waste, metabolic waste, and the efferent arteriole will take the blood to the body which has been already filtered. Okay, so this is called Malpighian corpuscle with Bowman's capsules and glomerulus. Now here you can see the Malpighian corpuscle, this part which has got the Bowman's capsule wall and the glomerulus inside, right? So that is the Bowman's capsule. So this is the part of the nephron called Malpighian corpuscle, including glomerulus and Bowman's. The main filtration, the process of filtration that takes place is this part called the Malpighian corpuscle. The blood vessels going inside, innervating into the glomerulus, where the metabolic waste or nitrogen waste is brought into this, and there is a filtration of the waste, and that goes as filtrate into the tubular part, or you can say renal tubule. Okay, where we saw that there is a distal convoluted tubules and loop of Henle and the collecting duct. So this is the collecting duct. This part is the collecting duct. Okay. Now you can see the glomerulus, the Bowman capsule, and then is the proximal convoluted tubule. Okay, and then the loop of Henle, where the reabsorption of certain substances takes place. And this is important because what happens during the formation of this filtrate, some of the important substance needed for the body is also goes along with the filtrate, that is reabsorbed back into the blood with the help of this part called loop of Henle. Okay, so there's a distal convoluted tubule. After this loop of Henle is the distal convoluted tubule, and then the collecting duct. Once the liquid that enters into the collecting duct, that is the fully formed urine which comes to the collecting duct, distal convoluted tubule, DCT, and then the collecting duct. Then ultimately it goes to the urinary bladder via this collecting duct. Okay, a ureter, via ureter, right? So proximal convoluted tubule, one, number that's one, shown in the green color, and then is the DCT or distal convoluted tubule. Loop of Henle, you can see the thinner. It is very thin, and that's the reason it's so easy. Reabsorption can take place the wall of this through the wall of this loop of Henle. Okay, so number two. Number three is this distal convoluted tubule, right? Now coming to the collecting duct, this is going to join the ureter. So collecting duct is actually carrying the fully formed urine, okay, and that is going to join this ureter, then going to urinary bladder. Okay, so temporarily for some time the urine is stored in the urinary bladder, and then it is released through the urethra. Okay. Now what happens here? Bowman's capsule is there, and then we have got the blind end of the renal tubule forms a double-walled cup-like capsule. They call Bowman's capsule around the glomerulus. Once this efferent arteriole, the blood vessels coming and they're forming a network, some network-like a structure. Okay, and these are branched also, and these are actually going to the blind end, which is cup-like capsules. That is Bowman's capsules. And that is together it's called the Malpighian corpuscle. Okay, the glomerulus. The blind end of the tubules forms a double-walled cup-like Bowman capsule which surrounds the glomerulus, as you can see. So this capsule is surrounded, double-walled capsule is surrounding the glomerulus, which is actually the network of the branch network of the blood vessels. Okay, so now this is the more detailed, you can the enlarged view. Okay, macula densa is the one layer of the tissue that is present in the we can say Malpighian corpuscle, or you can say near the Bowman's capsule. Podocyte. Okay, there is another kind of cell. Bowman's capsule, you can say within the capsule, this is the glomerulus, and there is Bowman's space capsules with the space. Okay, and then the proximal tubule starts from the one end. So at the other end, there is the blood vessel coming into the Bowman's capsules with the macula densa, double-walled cup-like blind end of renal tubule. You can see this one. Yeah, this is the from where? On the outer parietal layer of squamous epithelium. So this is important for the exam. Outer parietal layer is made of the squamous epithelium in case of the Bowman's capsule. Outer layer, okay, or outer parietal layer of the Bowman capsule is made of the squamous epithelium. The inner wall of the Bowman's capsule has certain unique cells called podocytes which wrap around each capillary. So that is the another point. Each inner wall, okay, has got certain unique cells called podocytes which are present around each capillary. So these podocytes are near the just surrounding these capillaries. Okay, it's a unique kind of cells, and these inner visceral layer you can see with the podocytes. The Bowman capsules and glomerulus, as I told you, is from the Trunkie and corpuscle. The podocytes are arranged in an intricate manner so as to leave some minute spaces called filtration slits or slit pores. Like you observe in the case of the blotting paper or kind of filter paper, there are some kind of pores through which the liquid or the solvent or the filtrate can pass through. Okay. Similarly, here also these podocytes are arranged in such a way, in an intricate manner, that leaving a small space or called slit spores. Okay, so that the filtration of the metabolic waste can take place as the filtrate. Okay. So endothelial cells of the capillaries have numerous pores on the or fenestrations. Okay, endothelial cells of the capillaries and have the numerous pores. Even the capillaries, okay, the endothelial cells, they have also got some numerous pores. So here you can see this part is the glomerulus, okay, and this one is the tubule. The tubule from there is one blind end, and you can see this one. The glomerulus, along with the Bowman's capsule, constitute the Malpighian body or Malpighian corpuscle. The Bowman's capsule and the glomerulus, you can see there's a tubule on the other side. Okay, when find, okay, now this one, glomerulus, and this is the afferent renal arteriole, and that is the efferent renal arteriole. Afferent is bringing the blood for filtration. Okay, the Bowman's capsule encloses a tuft cap of capillaries. Okay, the network of capillaries, branched capillaries, called glomerulus, formed by the afferent renal arteriole, a fine branch of renal artery. Okay, so that is the glomerulus, how it is formed. Now renal artery, you can see this one, the renal artery, which is going to branch into capillaries, and then it goes into the Bowman's capsule as glomerulus. Afferent arteriole, the coming out. Okay. Then you can see the glomerulus, efferent arteriole, renal artery, the main, okay, which is going to be branched into the glomerulus. Then the efferent arteriole is the outer one, which is taking the blood towards the other parts of the body. Okay, so here you can see the Bowman's capsule along with the glomerulus. Blood from the glomerulus is carried by an efferent renal arteriole of a lesser diameter, because the metabolic waste is removed, it is filtered, so the lesser quantity of blood can move, okay, are moved from the efferent arteriole towards other part of the body. Okay. Now this glomerulus along with the Bowman's capsule is shown, and you can see the tubular part. As I told you, the renal tubule has got 4 parts: proximal tubule or proximal convoluted tubules, and then is the loop of Henle, okay, and then is a distal convoluted tubule and the collecting duct. Okay, so renal tubule is this one, and then now glomerulus, you can see the Bowman's capsule. Number 1 is already shown in the previous slide also. Proximal convoluted tubule, PCT, then loop of Henle, okay, then is the distal convoluted tubule, DCT, okay, and then is the collecting duct finally. Okay, so glomerular collecting duct, okay, CD, collecting duct, right? So cortex and medulla. Now this is an important point to be noted. Yeah, this is important exam also. This part, the line which is shown in the black color, straight line, above that line, that belongs to cortex. Okay, that is the outer side, which we observe cut the kidney into 2 halves, or we see the section of the longitudinal section. We observe that there are 2 distinct regions can be observed. Okay, and with the help of these we can understand that the outer one is the outer cortex and the inner one is the medulla. So this loop of Henle and the part of the collecting duct that is actually constituting the medulla, inner medulla. Okay, so inner medulla consists of loop of Henle and the part of the cortex, and also some yes, that loop of Henle and the collecting duct. Okay, so here Bowman's capsule is the proximal convoluted tubule. The tubule continues further and forms a highly coiled proximal convoluted tubule. Once it will start, okay, as a tubule from the Bowman's capsule, that forms a highly convoluted coiled proximal, and that's why it's called convoluted tubule, PCT. Then is the PCT continues a hairpin shape, very fine, thinner, okay, loop of Henle which has descending and ascending limbs, okay, and you can see that the descending limb is connected to the PCT, and as descending and as the ascending limb is connected to the you can say the this one is the DCT, distal convoluted tubule, right? So here is a hairpin turn, then the descending limb. Now proximal convoluted tubule, proximal part of the ascending limb is thin and the distal part is thick. Okay, so this is the feature characteristics of this. The proximal part of the ascending limb is thin, okay, but is thin, and the distal part is thick. Ascending limb, this is ascending limb, and you can see this is thin, okay, thin and thin. The descending limb, convoluted tubule is there. The thick this ascending limb continues into distal convoluted tubule. So this is thick. So first it is thin, then it is thick, right? So ascending limb and ascending limb thin, descending limb thick, right? Okay. Now we will see this distal convoluted tubule here and the collecting duct. This distal convoluted tubule continues as the initial collecting duct in the cortex. So distal, that is the initial collecting duct for the part of the distal collecting tubule that starts it continues as the initial collecting duct, and then it moves that it merges into the main actual collecting duct, right? So some initial collecting ducts unite to form a straight collecting duct which passes through the medullary pyramid, okay, medullary pyramid, like I showed you yesterday. Distal convoluted tubule and collecting duct, you can see. So these are the parts. So these two parts are going to merge into the collecting duct. So this part is the initial, okay, right? So this is the part which is going to this part. So this is here. So this one, this one, okay. So this is the now going to meet the collecting duct, and then it forms. So in the medulla of the tubes of each pyramid join and form the duct of Bellini. So this is important. Duct, this is medulla which finally opens in the tip of the renal papillae, which I showed you yesterday. Duct of Bellini opens on the tip of the renal papilla and into a calyx that we discussed yesterday, right? So the contents of the duct of Bellini are discharged into the renal pelvis through the renal calyx, okay, and then it goes to the collecting duct. Finally it goes, no. So afferent arteriole, this is the Bowman's capsule, then the glomerulus, then the peritubular capillaries. Peritubular capillaries are present. The efferent arteriole emerging from the glomerulus forms a fine capillary network called the peritubular capillaries around the renal tubule, because this is carrying the already the filtered blood, and which is going into the different parts of the body, right? Okay. The blood from the glomerulus is carried away by an efferent renal arteriole, carrying away. And efferent arteriole is bringing, afferent arteriole is bringing, and efferent arteriole is carrying away, right? Glomerulus. So diameter of efferent renal arteriole is lesser, as I told you earlier. It is smaller in size, thinner. Okay, so diameter is lesser as compared to the diameter of afferent arteriole. So efferent arteriole, peritubular capillaries, the vasa recta. Okay, that is the portion of peritubular capillaries that surrounds the loop of Henle is called vasa recta. So this is important, because when the reabsorption of the substances takes place, that needs to go into the blood. So this is there is a kind of some blood vessels surrounding the loop of Henle. So vasa recta is the blood vessels which is surrounding, okay, it is highly reduced or absent in cortical nephrons. Vasa is well developed in juxtamedullary nephrons. Okay. The nephrons are of two types. There are cortical nephrons and juxtamedullary nephrons. Now we'll see what is this. The cortical nephron is the majority of the nephrons are cortical nephron. Many are, okay. And cortical nephron means in the majority of the nephrons, the loop of Henle is too short and extends only very little into the medulla. Okay. So as I told you, the loop of Henle part, it belongs to the inner medulla, and you can say that here in majority of nephrons, the loop of Henle is too short, okay, and extends only a few and very little into the medulla. So that we can absorb here. You can see where we do not find the vasa recta. So that is the case. Okay. So cortical nephron where we do not find vasa recta is big, prominent, distinct in case of the juxtamedullary nephron. In case of juxtamedullary, the loop of Henle may be long. Some of the nephrons are juxtamedullary nephrons, and in case of juxtamedullary nephrons, in some of the nephrons, the loop of Henle are very long in this type, okay, and run deep into the, and then running and going inside deep into the medulla. Okay, that means, so these are the two distinct. This is the important for the exam: juxtamedullary and the cortical nephron. Okay, and the cortical nephron are shorter inside. Okay, but juxtamedullary are longer with the long loop of Henle going deeper into the medulla, inner medulla. Right. So this is the juxtamedullary nephron. Now, if you observe and go through this, the part of nephron, you can see the that is the Bowman's capsules. Okay, there is the epithelium called simple squamous epithelium. Now, in many times I have seen, and different exams, these questions are asked: what type of epithelial tissue is present? In case of Bowman's case, it's a simple squamous epithelium. In case of PCT, proximal convoluted tubule, it is simple cuboidal with brush border. Okay, simple cuboidal. Okay, and with brush border. In case of descending limb of loop of Henle, it is simple squamous epithelium. And in case of this part, the thin segment of ascending limb of loop of Henle is a simple squamous epithelium again, and thick segment of the ascending loop of Henle, it has simple cuboidal. Just remember this. In case of ascending, both descending and ascending thin, it is simple squamous, but it's ascending thick, it is a simple cuboidal. So this is the critical question, important. Okay. So many students, they are confused. So please remember this. In case of the thick part of the ascending limb, okay, that will have the simple cuboidal, not simple squamous. In case of distal convoluted tubule, simple cuboidal and with brush border, without brush border. Okay, that like we have seen in case of the PCT, it was with brush border, but not in DCT. So character, cortical nephrons and juxtamedullary nephron, we will compare this one. Number majority 80% are the cortical nephron, and juxtamedullary are only 20%. Location of Malpighian body, the superficial cortex and inner cortex close to medulla. So these cortical nephrons are present towards the superficial region. Okay, when you cut the longitudinal sections, you will find they are in the upper region, and these juxtamedullary nephrons are towards the medulla, inner, close to medulla, right? Loop of Henle short in case of cortical nephrons that I showed you in a slide, right? So in case of the juxtamedullary, these loop of Henle are long, running deep into the medulla, right? Vasa recta are absent or highly reduced in case of cortical nephrons and present in case of juxtamedullary. Okay, so now I'll go through some questions. The structural and functional unit of kidney is the Malpighian tubule. Number B is the nephron, or it is the renal tubule, or the glomerular. It is the nephron. Okay, this forms the basic structural and the functional unit of kidney. The Malpighian body is formed by Bowman's capsule, glomerulus, both 1 and 2, or renal tubule, both 1 and 2. As I told you, Bowman capsule and the glomerulus. Initial collecting duct is formed by descending limb, ascending limb, DCT and PCT. So initial collecting duct is formed by DCT, right? Distal convoluted tubule. So they are going to join the collecting duct. A glomerulus is formed by renal tubule or the afferent renal arteriole or efferent renal arteriole or PCT. It is the afferent renal arteriole which is bringing the blood for filtration. Okay. Loop of Henle is too short in cortical nephron. Yes, it is in medullary nephrons or juxtamedullary nephrons or all. It is very short in case of the cortical nephron. Okay, and long in case of juxtamedullary nephron. Right. Peritubular capillaries around the renal tubule are formed by afferent renal arteriole efferent renal arteriole or loop of Henle. So here you can see it is the efferent renal arteriole is the one that forms the peritubular capillaries, because when this is taking the blood away, there are peritubular capillaries, okay, are taking the blood away from the kidney, right? So this was the all about the structure of nephron, okay. So what we discussed in this was about the structure of the nephron. The structure nephron being the structural and the functional unit of this kidney are responsible for the formation of urine. There are two distinct parts, as I told you yesterday, external medulla and the inner external cortex and the inner medulla, and these nephrons are very minute microscopic structure with some distinct parts, that is the Bowman's capsules and the glomerulus. Bowman's capsule along with the glomerulus called the Malpighian corpuscles. These Bowman's capsules are double-walled, thin, double-walled, and they are having the supply of the renal arteriole as efferent renal arteriole branching into the capillaries, a tuft of capillaries into the which are bringing the blood for filtration. Okay. Then there is the proximal convoluted tubule arising from the one end and is highly coiled, and then it forms after extension into a loop of Henle, which is very thin, and there are two limbs, descending limbs and ascending limbs, as you see in the picture, and this ascending limbs joins the distal convoluted tubule. Then distal convoluted tubule joins the collecting duct, and this collecting duct is the actual receiving the fully formed urine and going to join the ureter and then to the urinary bladder. Okay, so this is how the different parts of the nephron is there, and we can see there are initial collecting duct formed in the distal convoluted tubule, which joins the collecting duct ultimately, right? So these are the different parts of the nephron which plays a very important role in filtration and formation of urine. Okay. There are two kinds of nephron, that is the cortical nephron and the juxtamedullary nephron. Okay, and in case of the cortical nephron, the loop of Henle is very short. Okay, and in case of juxtamedullary, there is the vasa recta. In case of cortical nephron, vasa recta is very reduced or absent, but in case of juxtamedullary, vasa recta is present, and you can see here lightly this here in this picture in comparison, right? Okay, so the dashed line you can see the comparison, right? So that one, the A and B, the juxtamedullary nephron are long, okay, big, longer in length, longer in size, and you can see in case of cortical nephron, but this cortical nephron is much more in number. Majority of these nephrons are cortical nephron and only 20% are juxtamedullary. That means 80% are cortical nephron. And in case of juxtamedullary nephron, the loop of Henle is long, innervating, going or extending deeper into the medulla. Okay, so this was all about the structure of nephron, and so maybe in the next class we will study some other topic, and maybe the filtration or filtration and formation of the urine. Okay, so thank you for joining and listening today. Okay, and we'll study again in the next topic next time. Okay, so thank you for joining. Goodbye.